If you like this video, please subscribe. Thank you. Nicotinamide adenine dinucleotide phosphate is an important coenzyme derived from nicotinamide or vitamin B3 from the diet. It exists as an oxidized form, NADP+, or in a reduced form, NADPH. NADPH has several important and unique uses in a number of biological reactions, which will be explored in this podcast. There are two major sources that produce NADPH in the body. The first one is the hexose monophosphate pathway, or the pentose phosphate pathway, which produces two NADPHs every time a molecule of glucose 6-phosphate is oxidized. This is the major source of NADPH in the cell. A second source that produces NADPH in the cell is Malic enzyme, which converts malate to pyruvate and CO2, and that enzyme produces NADPH as well. NADPH is an important coenzyme required for at least 5 distinct reactions. The first one is the regeneration of glutathione by glutathione reductase. The second use of NADPH as a coenzyme is by NADPH oxidase, which is an important component of the immune response. The third important use of NADPH is by the monooxygenase system of the liver, the cytochrome P450 dependent system that introduces hydroxyl groups into organic molecules. The fourth important use of NADPH is in the reductive biosynthesis of both fatty acids and cholesterol. The fifth important use of NADPH as a coenzyme is by nitric oxide synthase, which produces nitric oxide. The first important use of NADPH is the regeneration of glutathione from oxidized glutathione and by a glutathione reductase. Glutathione produced by glutathione reductase is needed for detoxifying hydrogen peroxide by glutathione peroxidase. Each time a molecule of hydrogen peroxide is detoxified or converted to water, a molecule of glutathione is used up by glutathione peroxidase. It is noteworthy that glutathione peroxidase is especially important for red blood cells, which have a high oxygen concentrations that will produce spontaneously high amounts of hydrogen peroxide. Detoxification of hydrogen peroxide in red blood cells is an important protective mechanism for red blood cells to prevent oxidative damage caused by hydrogen peroxide. The regeneration of glutathione by glutathione reductase and NADPH ensures that glutathione is constantly available as a coenzyme for glutathione peroxidase involved in removing the toxic effects of hydrogen peroxide on red blood cells. The second important use of NADPH is by the enzyme NADPH oxidase inside leukocytes or white blood cells. This constitutes the initial step in what is designated the oxidative burst inside white blood cells as part of the immune system's response to invading microbes and other pathogens. NADPH oxidase will produce superoxide and other reactive oxygen species that will be responsible for attacking invading phagocytized pathogens within the leukocytes or white blood cells. It is important to note that the oxidative burst within leukocytes is one of two components used by the immune system to attack or destroy phagocytized pathogens. The third important use of NADPH as a coenzyme is by the monooxygenase system in the liver. There are two monooxygenases in the liver. The first one, associated with the endoplasmic reticulum, is responsible for detoxifying ingested organic molecules by introducing a hydroxyl group to their structure. In doing so, it makes those organic molecules more easily water soluble and more easily disposed of. The second monooxygenase system in the liver is associated with the mitochondria and is quite important in the biosynthesis of steroid hormones from cholesterol. Both of these monooxygenases in the liver are cytochrome P450 and NADPH dependent enzyme systems. The fourth important use of NADPH as a coenzyme is in both fatty acid synthesis and in cholesterol biosynthesis. Both of these pathways require the reductive equivalents present in NADPH during fatty acid elongation, as well as cholesterol biosynthesis. The fifth important use of NADPH is that it is a required coenzyme for nitric oxide synthase, which produces nitric oxide from arginine. This enzyme requires NADPH as a coenzyme, and it will produce nitric oxide from arginine. Nitric oxide is an important second messenger produced in endothelial cells, which is in part responsible for smooth muscle relaxation and vasodilation. If you like this video, please subscribe. Thank you.